In today's video, we're going to be looking at proteins, which are the third biological molecule that we need to know about for AQA A-level biology. So proteins have many different functions within the body. For example, they can be involved in growth and maintenance of the body. So proteins are kind of considered the building blocks of the body, and it's very important that humans have sufficient protein intake. This is particularly important in childhood, where we see these periods of excessive growth. So it's really important that the protein intake by children matches the demand needed to allow them to grow sufficiently. And likewise, as you get older and you stop growing, you still need to maintain the body. So it's very important that there is sufficient protein intake to allow this. Proteins can also act as signaling molecules. So certain proteins act as signals by binding to cell receptors and causing a biochemical reaction to occur. Proteins can also act as cell receptors themselves. So, for example, G protein coupled receptors and receptor tyrosine kinase are protein-based cell receptors. And we don't need to know this in a lot of detail at all for A-level biology. It's just important to be aware of some of the functions of proteins. And they can also be involved in supporting muscle contraction, which obviously allows you to move. So proteins are made up of these amino acid subunits or monomers. So we have a look at the structure of them. There's the central carbon atom. Now, there are 3 groups that are similar between amino acids and 1 variable group. So if we look at the similar groups, amino acids all contain a hydrogen atom, an amino group, which is this NH2 group, a carboxyl group, which is this COOH group, and then they have a variable region which allows these amino acids to be different. So most of these variable groups contain carbon attached to some other molecules. So for example, if you had CH3 as your variable group, it would form the amino acid alanine. The only exception to this is glycine, where its variable group is just another hydrogen atom that is bonded onto this. So amino acids can bond through a condensation reaction, similar to what we've seen in carbohydrates and lipids so far. And if you get 2 amino acids bonded together, they form a dipeptide, and obviously if you keep on adding these subunits, you get a polypeptide, so multiple amino acids, and proteins are considered polypeptides. Okay, so now we're going to run through how to draw out an amino acid. Now obviously we have just seen its basic structure of this central carbon, our R group, our attached hydrogen, our amino, or sometimes called the amine group, which is NH2, and our carboxylic acid. However, we need to be able to draw this out fully because both the carboxyl group and the amine group are involved in forming a dipeptide. So let's have a look how to draw this. We've still got our central carbon. We'll still draw our attached hydrogen atom. And you may be given a variable group to draw out, and they may state this, and often you don't have to draw all the bonds. So let's say our variable group is a methyl group, which is CH3. You would just write CH3. Sometimes they may ask you to draw it out, in which case for this example, because carbon likes to make 4 bonds, we would do 1 hydrogen here, 1 up here, and 1 on this side. But often you were just expected to draw it as so. Or they may not give you the molecule that's attached to the variable group, and in which case you would just replace this CH3 with an R. But for now, let's leave it as CH3. So the next things we need to do is to be able to draw out our amino and our carboxyl groups in full. So let's start with the amino group. So normally we'd have a line from our central carbon and we would write NH2. This time we'd do an N. I like to do 2 sticks coming diagonally off to do my H's. You could do top and bottom. In some ways that's better, and you'll see why in a minute, but it's just personal preference. And then again, for our carboxyl group, we do a stick coming off on the right. We draw another carbon, and this time we have our double bonded oxygen and our hydroxyl group. So that is a full structure for an amino acid. Now let's take a look at how amino acids react to form dipeptides. So when we want to draw out the condensation reaction occurring between amino acids, obviously and most importantly we need to draw out our 2 substrates, which here will be 2 amino acids. Now I've just drawn generic ones here, so you can see that the variable group is just denoted by an R. However, you may be asked to draw out the specific amino acids, so they may say between glycine and alanine, so one of these variable groups will have a hydrogen for glycine and one will have a CH3 group for alanine. So you don't need to know the side groups that are required for variable groups until you get to degree level, so they will, or they should, give them to you. So don't spend time trying to learn them. It's pointless information. You can also see that I've drawn the bonds on my amine and my carboxyl groups slightly differently. Rather than diagonally, they're now pointing either vertically or horizontally. And this is because it just makes it much easier when we draw out this reaction. So the 2 groups that are involved when forming a dipeptide is the carboxyl group of one amino acid and the amino group of another amino acid. Now this is a condensation reaction, so we obviously know that water is going to be released, and our reacting groups are this hydroxyl group attached to this carbon atom on our carboxyl group of one amino acid, and this hydrogen atom attached to our amino group on our other amino acid. So what we need to do to draw our reaction, we need to draw a circle around the reacting groups. So that is these 2 groups here. And then normally if we had enough space, we would draw an arrow and draw our product. I don't have enough space on this screen, so this is how it would look for the first stage of our reaction. And rather than draw an arrow, I'm just going to rub out the relevant bits and show you the products. So let's get rid of this hydroxyl group and this hydrogen group, because they will be lost in our reaction. And now we would get a bond forming between this carbon from our carboxylic acid group from one amino acid to the nitrogen in the amine group or the amino group from our other amino acid. This is a very long bond. Normally you would not need to draw it this long. It would look more like this one above, and then you'd draw the other attachments. But for the sake of space and not redrawing them, this is how I'm going to draw it. So obviously we need to label our bond. So we'd write peptide bond because that is what forms. I'm just going to put P because of space. And because it's a condensation reaction, we've obviously lost water. So we need to denote that, plus H2O. And so those are the products of our condensation reaction. We've got our dipeptide. Now, as I said earlier, if you were given specific dipeptides to draw out. So I believe the ones I gave an example of were glycine and alanine, and these have now formed a dipeptide. You would just label it dipeptide. You don't need to know what the dipeptide is. I've done a degree in biology and I don't even know what the dipeptide is. It's completely irrelevant. So you just label it dipeptide. And obviously, because this is a condensation reaction, we can do a hydrolysis reaction to split our dipeptide back down into its amino acids. So this time we have our H2O. We draw an arrow with it going towards our peptide bond, because remember this is a hydrolysis reaction. Hydrolysis meaning water and split. The water molecule is going to split our bond. And for stage 2 of the reaction, again we'd have the arrow pointing to our peptide bond and our water molecule to show stage 1 of our reaction. And then we would normally draw an arrow to show the products. I don't have enough space, which is why it is drawn as so, and why I'm rubbing things out. And then, because obviously water split this bond, water is going to be split up, and it's going to attach so that we have a full carboxyl group here and a full amino group here, and we have got our 2 amino acids. Now again, if the question just asks for generic amino acids, you would just label them amino acid. Excuse my horrible handwriting. But if they give you the specific amino acids, you must label them specifically. So the example I've continuously used for this question is glycine and alanine. So you'd have to label them specifically, and they could give you any amino acid. They could give you cysteine, proline, anything like that. So just make sure you read the question very carefully, and maybe the best thing to do would be to highlight the names if they give you a specific amino acid. So now let's take a look at protein structure. Protein structure is split into 4 different levels, and these are primary, secondary, tertiary, and quaternary. So let's take a look at each of these. A protein's primary structure is simply the amino acid sequence that forms a polypeptide chain. So we can see this on the image on the right hand side, and this is essentially, we've just seen how a dipeptide forms via a condensation reaction. Well, if we continue to add amino acids through further condensation reactions, eventually we'll get this polypeptide chain, and this chain determines the protein's final structure. So then we move on to protein's secondary structure, and this is where this polypeptide chain begins to fold and hydrogen bonds can form between amino acids, and this can form either an alpha helix or a beta pleated sheet. So if we look at the image on the right hand side, we can see an alpha helix is this very coiled structure, almost like a spring or a slinky, if anyone had one of those when they were a kid. And on the right, this yellow structure is a beta pleated sheet. These are a little bit more difficult to describe their structure, but they almost form, if you look at the first image in the primary structure one, you can see it almost sort of zigzags. So they fold into these zigzags. The best way I can explain it is if anyone made those cut out paper people that all held hands when they were younger. When you fold out to get your little chain of people all held together, when you're unfolding it, that's almost like the structure of a beta pleated sheet. So then proteins fold further, and this is their tertiary structure. So further folding allows formation of more hydrogen bonds, and we also get ionic bonds forming. And ionic bonds is a bond formed between an area of positive charge and an area of negative charge. So some amino acids have a positive charge, and these can interact with amino acids with a negative charge, causing our chain to fold even further. And we can also get a special type of bond called a disulfide bridge or a disulfide bond, and this is a bond that forms between two sulfur atoms. So the amino acid cysteine is the only amino acid that contains sulfur, and if we have two cysteine residues close together, these can form a disulfide bridge. So we can see on the image on the right, we've got this further folded chain that contains both alpha helices and beta pleated sheets, and these two structures can be found within the same polypeptide chain. So it depends on the amino acids that are present as to what they form, and to how long they are as well. And the final level of structure is the protein's quaternary structure. And this is essentially where multiple polypeptides assemble and they form a larger structure. So a really good example of this is hemoglobin, which is 2 alpha and 2 beta chains. And they form together and they create this larger structure. So we can see on the right hand side there that we've got 4 different coloured, almost like blobs, and these represent the individual polypeptide chains. So now let's talk about some of protein's functions. I covered this briefly in the first slide. However, these are the really important functions of proteins that you will need to know and know details of for your exams. So their first function is as enzymes. Hopefully we all remember what enzymes are from GCSE biology, but just in case we don't, enzymes are proteins that have roles in either breaking down molecules into smaller molecules, or joining molecules together, or changing the shape or the properties of some molecules. And so all enzymes are proteins. But not all proteins are enzymes. It's a little bit of a confusing thing to get your head around. All enzymes are proteins, but not all proteins are enzymes. So enzymes are roughly a spherical shape, and this is because of really tight folding, and this allows them to have their specific active site to bind to their substrate. Enzymes are also soluble and they have roles in metabolism, so they're needed to break down food, and like we just said, they can synthesize these larger molecules. Proteins also have structural roles, so they can provide physical strength, and this occurs through the cross-linking of polypeptide chains. So they lie down in parallel and they cross-link, much like cellulose. If you don't remember about that or haven't covered that yet, have a look at my video on polysaccharides. But essentially cellulose is made up of chains of beta glucose, and these cross-link between them and provide a strong and rigid structure for plant cell walls. It's the same with proteins here. They can cross-link with these parallel chains and perform these and provide these very strong structures. So structures like collagen and keratin is the substance that's found in your hair and your nails. So proteins can also form antibodies, and these are really important within our immune response. And antibodies consist of 2 light and 2 heavy chains, and they also contain a variable region, which makes them specific to particular antigens. So we can see the structure of an antibody on the right-hand side. It's this Y-shaped protein, and we'll have a look more at antibodies and the immune system in a later topic. And proteins can also function as transport proteins, and so within cell membranes we often see these channel proteins which allow transport of ions across cell membranes. And certain amino acids are hydrophobic, which means they dislike water and they don't want to interact with it, and we can get hydrophilic amino acids, which are water-loving amino acids, so they want to interact with water. And much like we saw with droplet formation in triglycerides, again, if you've not seen this or forgotten about it, check out my video on lipids. But much like droplet formation with triglycerides, where the hydrophobic fatty acid tails form inwards and form the inside of the droplet with the glycerol heads facing outwards because they're hydrophilic. Proteins can act in a similar way. So there's folding to conceal hydrophobic regions on the inside of a structure and have hydrophilic regions on the outside and interacting with water. And this almost forms this pore-like structure and allows ions to transport across the membrane. So how do we test to see if a protein is in solution? We use something called a Bioretest, and this is the last test for biological molecules that we need to know, and it's a Bioretest. So this test is split into 2 stages, and the first thing we need to do is to make the solution alkaline. So we add sodium hydroxide solution, and for anyone who's studying chemistry, we know that hydroxides are alkaline, so they are a metal attached to a hydroxyl group, so an OH group, and when they are exposed to water, they split, or when they're in solution, they split into their positive metal ion and this negatively charged hydroxide, so OH-, and this is what makes our solution alkaline. So after that, we add copper 2 sulfate solution, which is this nice blue colour. And if a protein is present, the solution will change to a purple colour. If there's no protein present, then it will just remain blue. So let's recap what we've covered today. We have learnt that amino acids contain a hydrogen atom, a variable group, and this determines which amino acid they are, an amino group, which is our NH2 group, and a carboxyl group, which is our COOH group, and these groups are all around a central carbon atom. We've learnt that protein structure is split into primary, secondary, tertiary, and quaternary. So we start off with a polypeptide chain. This folds to form alpha helices and beta pleated sheets. Then there's further folding and formation of hydrogen, ionic, and disulfide bonds. And eventually we move on to quaternary structure, where multiple polypeptides can form this larger structure. We've learnt that proteins have roles in structure and transport, as well as forming enzymes and antibodies, and we've learnt that a Bioretest is the test needed to find out whether there's protein in solution, and this is performed by adding sodium hydroxide to make our solution alkaline, then adding copper 2 sulfate solution, and a positive result is indicated by a purple colour. So now that we've covered this topic, it's important to go back several times, make sure you fully understand everything, and then have a go at answering the following test questions. So number 1, describe how a dipeptide is split into 2 amino acids. Use diagrams alongside your explanation. Number 2, explain and give details of 2 functions that proteins have. And number 3, how is a Bioretest performed and what colour indicates a positive result? That's all for this video. Thank you for listening.